Good afternoon. I'm most pleased to present to you Dr. Sedat Nizamoğlu, who is one of our own. Sedat has graduated from Bilkent University with his electrical engineering BS degree, and then he continued in the physics department for his master's degree, and then finally had received his PhD in EEE from Bilkent. And once he completed his studies in our research group, he received a fellowship from Harvard University, and at Harvard he received a second fellowship as an award, and then he returned to Turkey, and currently he's at Koç University at electronics engineering, and he is at the moment running a DRC program. For Sedat, of course, I can tell you more because we worked very closely for many years, and we get to have our close contact over the years. And Sedat is one of those really patient scientists, and I'm sure over the years, sometimes my impatience for results push him further, but I'm always most impressed with this very calm, patient, and very friendly attitude. Without further ado, Sedat.
Thank you so much. Thank you so much for the very kind invitation and also introduction. During my PhD at Bilkent, I had a great time here and working with Volkan and with other professors here. Therefore, it is also a pleasure for me to come back again to Bilkent University. Today I will present you a topic on biomaterial-based photonic devices, and I will explain to you, in fact, unlike these laser pointers, in fact, we can use biomaterials or living entities even to make some lasers, LEDs, or some other photonic devices as well. So that I would like to open your horizon to a different material type, and so that we can do some optical tools and photonic tools by using another class of material. And today I selected three main examples. The first one is that I will talk about is biodegradable wavelengths. This is the first one that we can use for medical applications, which I will show you in this talk. And the second topic is lasers. In fact, we can do lasers by using biological cells or by using a cancer cell, so that we can use it maybe for in the future for therapeutic applications. And the third one is about LEDs, so that we can use in fact proteins to make LEDs as well. So that initially I would like to give you the idea basically, okay? Why do we use biomaterials? The main idea is this. I'd like to show you the main idea. So that let's say we have a device that we can put into the body, and this concept is as the device that we place into our body. And after we put this device into our body, then it will be in the tissue medium in our body. And this water mimics the tissue medium in our body. So that after we put this biomaterial-based safe device, which is also biocompatible with our body, after we put it, then it will degrade safely into the body after it finished its function. So that this is the main idea to make, in fact, implantable and biodegradable in vivo devices. And the first part of my talk is based on this idea, putting a functional material into the tissue and then using for medical application. And as we know that the light has a limited optical penetration there into the body, here on the left you see a Monte Carlo simulation, and And as you also see on this simulation, light can only penetrate a few millimeters in depth into the tissue. And as you can see on the right, after 2 millimeters, let's say at green wavelength, the number of photons that you can detect in the tissue becomes almost negligible. And one possible way to penetrate light into deep inside the tissue is that we can use an optical fiber and embed into the tissue so that at the targeted position we can deliver the light. However, after we use these conventional optical fibers, since these are made of plastics or glasses, they need to be very quickly removed from the body. And as a solution to these conventional device problems, we offer the solution using biomaterial-based devices, so that, as I showed in the video, we can put these devices into the body so that they will do their function, their job, and after they finish it, then it will degrade in the body safely, so that it will not disturb our health as well. And why biomaterials? Because they are biocompatible. Their biodegradation time can be tuned from minutes or years. Even we can use non-degradable polymers as well. Their stiffness can be adjusted. They can be made transparent, which we like so much as optics people. And also they are recyclable. At the same time, they are sustainable. For example, if you consider silkworm produces silk cocoons, and by using silk, we can make, for example, by using silk fiber from the silk, we can make transparent film as well from a natural source. Or synthetically, we can also synthesize in the lab environment as well. So that this is also sustainable technology, and also we can generate micro and nano scale structures on it as well. And as a first demonstration, as a first component that we can make out of biomaterials is optical fibers. And basically what we are, the technique that we are using is the fiber drawing technique. And it is a very simple technique as I showed here. Here we have a container, and in this container we have the biopolymer. And on top of you see a band slip. This is just a heater on top. We heat the biopolymer inside the container. And after it exceeds the melting temperature, it becomes liquid. And here the very thin line, I hope you can see it. This thin line is the melted liquid. And on top of a motorized pulley, we pull it. And by changing the carrier, the speed of this pulley, as we can see here, we can generate fibers from different diameters from 100 to 400 micrometer. And these fibers are basically made of biomaterial, polylactic glycolic acid in this example. And they are, as you can see, flexible, biocompatible, and at the same time they are biodegradable as well. And these optical fibers has a loss of approximately 0.5 dB per centimeter. And another material or another component that we can use for optical applications is the optical waveguides. And as you can see here, we started a biopolymer powder. And by using melt pressing technique, so that we heat in two plates and melt it, as you can see here, we can have this transparent and flexible biopolymer film. And then by using laser cutting technique, it is right now very frequently found in many academic settings, so that by using a laser cutting technique, we can generate any, for example, a waveguide shape, as you can see here, even any fan shape that you can produce by using this technique. And here you can see, for example, one waveguide example, so that the side walls are smooth. And by using the laser cutting technique on the edges, we can, as you can see here, we can generate some here micro bandings, so that we increase the extraction losses in these optical waveguides. Or if you increase the frequency of these micro bandings more, we can even increase more the propagation losses on these waveguides. So that this production also technique provides us as a flexible tool to generate any waveguide that we would like to see. Right now I will discuss what can be the benefit of using optical waveguide in a tissue. For example, here initially you see a simulation on the left, so that Monte Carlo simulation. On the left, we have a very external light source, so that which is focused on top of the tissue. And as you can see here, light can only penetrate a few millimeters in that. However, if we place a waveguide inside of the tissue, as you can see here, the light can penetrate effectively centimeters long, deep inside the body. And also experimentally, we confirmed it by using a wavelength mesh. We fabricated it. And as you can see here on top of a tissue, a few millimeters light can penetrate, but by using a wavelength inside the tissue, as we can see here, the light can penetrate much deeper, centimeters long. So that light delivery, what I would like to conclude is light delivery is not limited by the tissue scattering and absorption by using these biomaterial-based optical wavelengths. In principle, if you produce an optical wavelength or optical fiber, we would like to reduce the optical propagation losses. But if you design an optical wavelength for a medical application, then the design criteria can be different. Therefore, we analyzed the optical wavelengths in different environments, in air, water, and oil. And we produced polylactic, 100% polylactic acid-based wavelengths. And the height of it is approximately 240 micrometers. And by using the cutback technique in these environments, we look at the propagation losses. And on the x-axis, you can see the wavelength that changed from 0.6 millimeter to 1.4 millimeter. And in the air, for example, we start around 2 dB per centimeter, and the propagation loss decreases while we increase the wavelength due to the less number of bounces due to the reflection on the boundary of the wavelength. And as we can see here, while the refractive index increases, so that the propagation losses increases. And while we look into the oil case, as we can see, which has a refractive index which is close to the tissue right now, we start to have a very high significant loss, which starts from 20-30 per centimeter, and while we have 1.4 millimeter, the loss is significantly higher than 10 dB per centimeter. To decrease the losses, what we can do is, in fact, we can basically increase the height to 440 micrometer, so that with a wavelength of 1 millimeter, as we can observe here, then we can achieve approximately 10 dB per centimeter extraction efficiency. What this means, in other words, is so that we can dump the energy into the tissue 90% in 1 centimeter length. After we analyze the optical properties of these wavelengths, we look at their response in tissue environment. But before we look at its response in water, because the tissue is made of also water, so that the physical structure needs to be conserved in the tissue structure or water environment as well. And we look at, for different biopolymers, this is polyvinyl. It is a water-soluble biopolymer. And as we can see here, after, for example, 4 minutes, it starts to lose its shape. And after 10 minutes, it is already dissolved in the water. Instead of polyvinyl, we can also use silk fibroid. But what we observe is after 1 minute, as you can see here, the shape is not preserved. And also after 5 hours, it swells the water. So that it is also not a good choice to make optical wavelengths out of it. Here also we look at polylactic glycolic acid and polylactic acid, and they have a stable physical structure, so that we can safely use the wavelengths, optical wavelengths that we made out of these two materials. And therefore, we have chosen polylactic-based materials. And if you go to, for example, to a doctor, and if the doctor makes a suture on your skin, the doctor in general says, for example, you don't need to come after 1 month to take the sutures from your body because it is degraded. It will degrade automatically in your body. And the main reason why it degrades is because it has a content of polylactic glycolic acid 50-50, which degrades in 1 to 2 months in the body environment. And therefore, we have chosen polylactic glycolic acid 50-50 to make optical wavelengths out of it. And to understand the biological response from a body, we inserted these optical wavelengths into a subcutaneous of a mice, under the skin of a mice, and look at the degradation properties of the optical wavelength. And as you can see here, after 6 days, the wavelength still conserves, keeps its transparency, which is good to have an optical communication inside the body. And after 17 days, we start to observe opaque, which becomes opaque. And then after 35 days, it becomes fully degraded in the body. And the main reason, if we look at this degradation, is due to the hydrolysis of the ester bond. You can consider these polymers as long chain, okay? And due to the hydrolysis, you start to cut smaller and smaller pieces, this biopolymer chain. And we look at molecular weight, as you can see here, while the waveguide is in the body, because of these chain length shortening, as we can see here, the molecular weight decreases here while the days passes. And we observe a full degradation after 1 month, approximately. And here also one important thing is, one possible question is, why do we observe an opaqueness here? And the main reason is because the degradation mechanism in vivo happens on the surface and also in the volume as well. Because of this volume degradation in the waveguide, it generates discontinuities in the refractive index, and because of that, we start to observe this opaqueness start to be opaque. And so that what it shows is, in fact, polylactic or glycolic acid 50 is a biocompatible and biodegradable option to make the optical waveguides. And after we designed our optical waveguides, right now I will explain you the one possible application that we can use these optical waveguides. It's called photochemical tissue bonding. It's a very simple technique, and it is found at Harvard Medical School in 2002, and it is at clinical trials right now. And it's a very simple approach. What we need is an organic solution, a dye, which is an FDA-approved dye, rose bengal. And what we need is a green laser source or LED. And so that, let's say we have an incision, we apply in between this incision the rose bengal, and then we close the wound, and that irradiate with light. Then, because of the covalent cross-link between the here the amino acids, they become attached. The two surfaces of the tissue becomes attached. And the PTB is a relatively new technique, and it has quite nice properties, so that it's a fast approach, so that in a few minutes you can close the wound by using this technique, and it generates a watertight seal. At the same time, it is non-inflammatory. And here you see the results on a human patient, so that on the right it is made of sutures and on the left it is PTB. It generates also minimal scarring, as you can see here, so that after suturing you have the scarring, so that aesthetically also it is a good approach as well. And like all the photomedical techniques, it has the basic limitation of penetration problem, as we can see here. This is a porcine skin, and it is 1 centimeter thick. And if we shine an external light source on top of it, the light can only penetrate a few millimeters in that into the tissue. As a solution, we developed this system so that we have a laser source, and it is coupled to a conventional glass fiber. And after this fiber, the light energy in this fiber is coupled to the biodegradable optical waveguide, so that we dump all the light inside the tissue, so that advantageously the deep tissue photoactivation can be possible by using this technique. And at the same time, since it is biocompatible and biodegradable, you don't need to remove the waveguide from your body. To activate a large area, we develop this waveguide bundle. Here we have an optical fiber connection. This is made of a glass optical fiber. Then we have a mechanical stress reduction part, since the patient will move while you do the suture. We have this part. And then we have light distribution part, so that the light is distributed homogeneously, and then transported each one of the waveguide. And here, the part that you see here is the part where we insert into the body, and the light is extracted in this part of the waveguide. And this is the clinical procedure that we developed, so that, let's say we have incision, then we apply the dye, then we insert the waveguide and illuminate the targeted tissue, and then we cut the outside part of it, and then the remaining part inside will degrade in the body safely. And so that this is the procedure again. We apply the dye on a porcine skin, we place optical waveguide, we illuminate. While we illuminate, a little pressure is a good idea to put, so that on both sides will be attached together as well. And we cut the upper part of the waveguide from it. And as you can see, by using this optical waveguide, the light can penetrate deep inside the tissue. And as we can also see, by the increase the light energy applied onto the tissue surface, the affected area, which is shown here, the bleached area, increases, so that we can effectively photoactivate the deep inside of the tissue. And so that it is also shown here, the dye density decreases due to photobleaching, which shows us the photoactivation, so that almost 95% of the dyes become salt activated in this tissue. And we also confirmed this technique, measured the tensile strength, so that how does the strength, the bonding strength increases. And as we can see here, we use the peel-off technique, and the bonding strength can be increased to 5 to 10 times. And right now I would like this, right now so far this technology that we developed is also selected by a TV series recently. And as some of you know, I talked with some of you and said that they are watching the White Rabbit project from Netflix. If you would like to watch it, it is in session 1, episode 1. Right now I will show you hopefully a short movie so that it shows we make a incision on the porcine skin. And on the left you see a regular conventional suture. And then this is the rose makeup like in the solution, so that we will place on top of the porcine skin that we put on porcine skin, so that all the tissue surfaces are covered. And then basically what we do is irradiation, okay? So that we place the optical wavelength, and then we turn on the laser and we illuminate. And it takes a few minutes, so that for a few minutes long, we activate the tissue so that all in depth of the porcine skin will be photoactivated. And after, right now he asks, kind of, it is like Star Wars is explaining this right now demonstration of using lasers for tissue bonding. And after a few minutes, as we can see here, it takes a few minutes, and then we stop the laser, and then just what we need is to cut the upper part so that the patient will be free to move anywhere he wishes. So that then the rest will be degraded in the body safely. So that after this we place it into the body, it will degrade automatically and safely in the body. Right now I will continue with the second topic on the living cell lasers. And so that I will show you how you can make a laser. This is a laser made of inanimate material, non-living objects, but in fact you can make also lasers from living materials, identities as well. And the first living laser is demonstrated in 2011 by at Harvard Medical School. And basically what they did was they used a cancer cell and then they genetically modified it. And this cell can generate green fluorescent protein inside. And they put into a resonator, a laser resonator, in 2 meters. And then they pumped with their 5 nanosecond laser pulses. And then this cell starts to generate laser light out of it. So that this was the first demonstration of generating laser in 2011, which is also covered in Nature Photonics as well. And however, there are some also problems in this new technology, so that they are using genetically encoded gain medium, so that you need to have a cancer cell line, and then you need to transfer it, incubate, and cell sort out. It takes around several days, 3-4 days, to have a cell that has sufficient GFP level inside. Therefore, it is in fact it is not convenient to use for for the applications that do not require any genetic encoding. And for this, we find out a simple solution for that one. So that in fact, the fluorescent molecular probes, if there are people here making some biological studies, they are very frequently using these biological markers in their experiments. And we use these markers because they are common in biology, common in biology. You can choose large variety of them, easy to apply on living cells, and at the same time you can detect very easily because their quantum yields are also good enough. And we choose among them the green cell tracker dye. And here you see a cell, cell here. And as we can see here, it generates a localized gain medium, so that after we stain it, we have just gain medium, the organic dyes just becomes active inside the cell. And also, it is right now much faster. For the genetic encoding, we need to wait a few days, 2 to 3 days, but by using this technique, it takes less than 1 hour. They have high visible quantum yield, even more than 50%. They have good photostability. And as you can see from these absorbers, we can also pump in the visible range as well. And they are also stable. After we stain the cells, as shown here, they become stable inside the cell. But if you use a standard biological organic dye, they diffuse out after they take the intake these dyes, so that these cell tracker dyes is also very stable at the same time and biocompatible, so that the cell viability is in average more than 98%, so that they can safely use these dyes as well. We did the same, we used the same structure, so that we take the cell, then we apply on top of the cell the organic dye, so that right now the dye is inside of the cell, this special dye. And then we put them into between 2 mirrors, which makes a laser resonator. And then we pump with 5 nanosecond long pulses to generate population inversion of these molecules. And then we observe lasing out of it, so that you see the phase contrast image of these cells. And this is the fluorescence image. And so that the fluorescence is everywhere inside the cell on the cytoplasm. And after it starts to generate cell lasers, what we observe is we only observe cell lasing in the central part. And the main reason is, in fact, if you consider that the mirrors are in the z-axis, let's say, so that, and we have a spherical medium, so that we can have the longest gain in the central part. This is the main reason, so that the gain can be larger than the losses in the central part. Therefore, we observe lasing in the central part of the cell. And the threshold energy was approximately 12 nanojoules. And as you can see here, the spectrum starts with a few multimode, and then as we increase the pump power to 240 nanojoule, the observed multimode laser emission becomes higher, as we see here. And the lasing threshold is dependent on the concentration as well. The optimum concentration is that we found out for cell tracker green dye is around 100 micromolar. And we also tested the biocompatibility of this approach. So that one question is, okay, you make a laser by the cell, but are the cells alive? Yes, but under some conditions. For example, on the here on this column, you can see, for example, for laser pulses, 140 nanojoule, and on the left you see the number of laser pulses that we apply on top of these dyes. And as we can see here, for example, this is 10 times larger than the threshold level, so that we increase the number of pulses, and we cannot observe any change in the cell morphology. And this becomes true until 7.5 microjoule. And while we start to apply around 1,200 pulses to these cells, we start to observe here some blebs, as you can see here, some small blebs, so that it shows that the cells start to die. So that what we can conclude is, in fact, the cells become alive until this threshold level, so that 2.9 microjoule, we can kind of safely use it. But if you go to beyond it, around, let's say, 7.5 microjoule, we start to observe the death of the cells. So that this is the safe region where we can generate cell lasers. At the same time, this new method also allowed us to look at cell lasers on top of adherent, so that for adherent cell lasers as well, so that they can attach, as you can see here, on top of a mirror. And we can also observe cell lasers on top of a mirror in between the resonators. And what is quite interesting that we found out is in free floating cells, in this one, in this example, as you can see, it has a spherical structure, so that it has a central symmetry. But if you have an adherent cell on a surface, then you start to perturb the symmetry on the system. And this kind of generates only some modes to oscillate in this cell, in the gain medium. And what we observe is, in fact, we can only observe, for example, the fundamental mode, 0, 0, or 1, 1, or their combination. So that, as we can also look at the spectrum, we can only observe single transfer mode laser emission. But if we compare it to the previous one, as you can see here, we can see multiple if we have a spherical free-floating cell. And therefore, what we conclude is, in fact, the cell right now function as a special filter, so that if we have a symmetric gain medium, and if we have a special filter on it, so that we kind of close some parts on the cell, so that the cell morphology shows a light behaves like a special filter, so that we can only see the observation of some lower number of modes, 0, 0, and 1, and get combination on these. But in the floating cells, we do need some hyperspectral imaging here, and we can observe a very high number of modes that oscillates in these lasers. And what can be the application of it? One application can be, we can right now in 2015, 2 years ago, by using this staining method and making lasers out of it, they demonstrated cell lasers, and they integrated beads, micro beads, to make respinning gallery modes inside the cell, so that right now the resonator is in the cell, and outside is the gain meter you can consider. And while the light circulates out of it, they demonstrated a cell laser, and they use it for observe the dynamic fluctuations on the cytoplasm, which is more sensitive than the current state of that. And another possible application is you can, since these are laser lines and they have narrow fluid half maximum, you can take thousands of cells by using this idea as well. And for the third topic, right now I will show you a protein integrated LED. As we know that the solid state lighting is an important phenomenon that affected our daily life. And one of the problems right now in our current sample is, in fact, we are using right now many electronic devices. And for example, this is a cell phone that most of us have had. And in general, we use maybe in 2 years and then replace it with a new model. And in fact, this is good maybe for us, but it is not good for nature. So that the average usage time, for example, for a cell phone is approximately 2 years. And even though their electronic lifetime is much larger than that, so that it generates a significant amount of waste in nature. And let me show you an island. This is a Pacific garbage patch, so that you can see right now islands made of these wastes as well. So that in our group, what we also try to do is we try to also transform the electronics to green material-based ones, so that after you use it, if you trash it, then they will degrade safely in the environment as well. So that what we are trying to do basically is like we do this transformation. Right now, maybe 40, 50 years ago, we had conventional plastic bags, which doesn't degrade in the nature. But right now, we have 100% degradable plastic bags that we can use. And we would like to use the same idea, so that we would like to make electronics so that are degrading after we use them. And there are some fundamental limitations in different materials. For example, phosphorus, it has a limited spectral tunability. It has a broad emission spectrum. And there are also concerns over the global supply. And at the same time, nanocrystals can be made from toxic heavy metals. And also there are some environmental concerns out of it as well. And as a proposed solution of using fluorescent proteins, in fact, as a replacement for these materials. Why fluorescent proteins? Because as we can see here, we can choose from blue to red region, and they have a narrow spectral alignment at the same time. Some of them can have high quantum yield, more than 50% as well. And here the nature, in fact, did something very interesting as well. Here you see the structure of a green fluorescent protein. For example, the first kind of quantum dot synthesis started with a core material, but nature around 1 million years ago, in fact, produced the core shell structures with high photostability. Here the chromophore is in the center, and here we have a barrel-shaped shield, so that it protects the emitting core inside of it. And at the same time, we can use bacteria to produce this type of fluorescent proteins, and it's biocompatible and edible. I looked at on the internet as well, even there are some sushis that we can use fluorescent proteins to eat as well. And we used to produce them E. coli, the bacteria. They can express, after the expression, we purify them. And here you see the confocal image, so that they produce GFP and cherry proteins, as we shown here. And it's a simple and effective expression system. And we collaborated with Photonic to produce these fluorescent proteins. It starts from transformation then induction, then cell harvest. And after cell harvest, the cells release their GFP into the solution, and they can be concentrate and purify them. And this is the final result. We have the eGFP and M-shape solutions that we can integrate for device applications. And if some of you see the spectrum for the first time for eGFP and M-shape, and so that for eGFP we can use a blue LED, for example, a blue light source to found it, and it starts to have emission around 520, 530s. And also for M-shape, as you can see here, it has an absorption around in the green region, yellow region, and then it has an emission in the red region. The quantum yields are not the best ones that we use, but there are also other alternatives that can use with higher quantum yield in the red region as well. And we did a very quick simulation out of it, and so that by using blue quant LED on top of it, eGFP and M-shape, and it shows that they can achieve high color random indices so that it can make a nice white light so that we can distinguish the colors nicely. And the spectrum is also highly efficient, so that if we have a green laser, let's say, a Dirac delta at 505 nanometers, it will have 683 lumen per watt. This is the efficient visible spectrum generation, but for this one, the efficiency can be around 400 lumen per watt, which is also quite good for a white light. And we use these fluorescent proteins for TV applications. If you look at a LED TV, for which are commercial right now available, it has a backlight unit made of white LEDs. And right now the current state of the art is they are using phosphor-based white LEDs. And instead of it, we, on top of this blue LED, we integrated eGFP and M-shape on top of it. And here you can see the color gamut that it generates, so that it has a 80% NTC color space. So that if you compare it with the conventional yellowish phosphor, it has a 68%. But the quantum dots, for example, is a better technology in terms of it. But if we replace this M-shape with the M-plus, this becomes also compatible with the quantum dot technology at the same time, so that the ATC color space can go up to 114% as well. And this is the first protein TV demonstration. So that right now, Samsung released the quantum dot TV, but maybe next generation, they may turn to fluorescent protein TV as well. So that it is right now a small TV, 3.5 inch, but just it is for proof of concept demonstration. Today I, in fact, explained to you some of the results on bio-integrated photonics, but we have also some efforts on neural interfaces, nanomaterials, nanophotonics, and optoelectronics as well. Here, and this is the key, right now we const of 2 postdocs, 6 grad students, and we have an undergrad student. And we have a quite interdisciplinary team. We have researchers from electrical engineering, material science, physics, and also he is from genetics. He is a third year student, but he is doing all the cell culture and biology related things. And I would like to also acknowledge the funding agencies as well, so that to conclude my talk, I discussed with you today the biodegradable waveguides, so that maybe in the future we may find some real-world application, or we may see them in clinical settings as well, maybe in the future, with biodegradable waveguides. And we discussed also the molecular probe-based biological cell lasers, so that we can make living lasers out of cells, and also fluorescent proteins that the nature generated for us we can also use for and integrate to optoelectronic technologies as well, so that the biomaterials hold a great promise for novel photonic applications. This is all of my talk. Thank you so much. Even though the nature generated is barrel shape shield as a shell material. He also observed the decrease, for example, I think, let's say for GFP, it decreases from 50 to 60% the quantum yield to 10%. And this is, I think, one of the main problems in this technology, like in quantum dots as well.
I thought very helpful.
It doesn't solve it extensively.
About this laser in the cell, you showed about the only changes about the morphology. What about the functioning of the cell inside the cell? If there is nothing happening about the morphology, how are you going to differentiate it?
Okay, you mean that how does the cell biology also continues?
Well, yeah, you told it, there is a threshold, but you only put that threshold in terms of morphology.
Yes.
But in terms of functioning, what's the threshold like?
Okay. For example, functioning, you mean, for example, as a neuron, you mean?
Let's say protein production.
Protein production, okay. Right now, there is a recent study, I think it was published in last 5 to 6 months. What they did was, they genetically encoded one of the cells, and these dyes, which are photo-bleached, is automatically regenerated inside of the cell, so that the cell laser can continue infinitely long run. What I'd like to say is, one of the limitations of these lasers is their stability, so that after you put into the laser cavity, then kind of this generates a photo bleaching on the, for example, proteins, fluorescent proteins, let's say. But they solved it by continuing the biological production of the fluorescent protein inside the cell, so that it regeneratively it can continue to produce the proteins inside the cells. So that I didn't put that maybe paper here, but they generated the first regenerative cell laser as well by that way. But I don't know exactly, there are some other functions maybe that are limited to neurostimulation or other things, but I think no one had any study on it as well.
So I'm just curious for the lasers. You use parallel ion lasers.
Yes.
Is there any reason we're not using elliptical mirrors or higher, not for that? The device works on the cell. Any device?
In fact, this is one of the, I think, the easiest way to get cell lasers, and this is therefore we use it. For example, as I showed here as well,
For efficiency purposes, maybe.
yes, it is very easy, because after you incubate the cells, you need to be a little bit quick as well, because they are from their natural environment. This is the sorry.
They actually viable.
Yes, for these parts, because for adherent cells, if they are not alive, they don't stick anymore on the surface. For example, and what they did was, for example, in other configurations, they used these whispering gallium or spectral beads, so that they used another resonator structure, so that it is possible to use other structures as well to make lasers out of biological cells as well.
Questions or comments? If not, I would like to thank Sedat Nizamoğlu.
Tamam.
Görüşürüz gençler. Gruplarınızı hazırlıyorsunuz. Bu toplantı
dezavantajları var tabii. Yani lazer yapmak belki bir avantajları olabilir. Şu anda bazı avantajları var. İşte fazla görmüyoruz. Evet, aynen. Mesela orada biraz altın
yiyecek falan gelsin.
yani memnun oldum bu arada. Siz de yani ne üzerine çalışıyorsunuz?
Haftaya mı oluyor? Cuma mı oluyor? Bir dersi var ya. Her cumartesi dersi. Ders olduğu zaman
Her cuma olunca diğerinde de oluyor büyük.
yani şey ders yani. Her hafta sizin bir şey gibi. Tamam bir şey almaz. Altıda kapatıyor.
Aynen.
Altıda kapatıyor. Hemen yetişiyor. Köfteyi bir olay işte gönderdim.
Nereye gitmiyor musunuz siz şimdi?
Ben bir saatten fazla zamanımız
Gidiyorsunuz. Tekrar bölüm söylese gidiyorsun gerçekten.
Ev yaptırdık.
Yani iki tane mantıklı şey konuşuyoruz.
çekmeye başlıyoruz. Teşekkür ederim. Yani bize bir anlamı olmayacak. Evet. Yani çünkü normalde kristalerde kuantum confinement var. Bu yani her şey orada bir şey oluyor. Yani orada
Ne sordu?
Bir daha da cevap almıyorum.
Ne sordu? İyi de
Farklı problemler, farklı bir şey.
ben istedim.
İleride de bulunma birisi var. Bir şeyler Bu arada iyi görmüş nasıl keyiflisin yani? Bir anda bir keyiflenme.
Azaldı işler. O yüzden
Nerede yatıyorsun canım?
şu an yatak geldi.
Geçiş olarak hayır yapıyor.
Aman tanrım.
Aslında hiç kimse tabii ben.
Aynen.
O da kitleyip bence anahtarını almıştır zaten.
Orada boş tamamen şu an. Orada bile gelmedi yani. Misafirlikten olmuş.
Kendisi içinde de kurulması lazım da tam denedik olmadı. Ya onların belli bir teknik sardı. Orada bir şey demek değil.